It is my pleasure to introduce you Dr. Rebecca Bart today. And Dr. Bart got her bachelor's degree in biology at the Reed College in Portland, her PhD degree in plant pathology at UC Davis, and her postdoctoral scholar at UC Berkeley. And in 2013, Dr. Bart began her own lab at the Donald Danforth Plant Science Center, where she is now an associate faculty member. Today, she is going to present her research, which focuses on CASSAVA and some important susceptibility genes that are encoded in this host genome and that are required by pathogens to complete its life cycle and consequently impact the production yield. So Dr. Bart, thank you today, and the floor is yours.
All right, thank you very much. Can you hear me okay?
Yes.
All right, great. Let me go ahead and share my slides and we'll get started. So it's my pleasure to be here today. I'd like to thank all of the people who have been organizing this seminar, and I am sorry that we didn't all get together in person. I hope you have a great time at the mixer this evening. I wish I could be there with you, but maybe next time. Before I jump into my research talk, I want to introduce quickly the institution that I'm at. It's a little bit unique, and I find that especially in these forums where there's a lot of trainees, people are often a little bit interested and curious and a bit confused about what the Danforth Center is. So the Danforth Center is a nonprofit plant science institute. We're located in St. Louis, Missouri, and we kind of try to bridge this gap between academia, traditional university settings, and industry. So we have one overarching governing mission, which is to improve the human condition through plant science. And in general, the work that goes on at the Danforth Center revolves around these three pillars: feed the hungry and improve human health, preserve and renew the environment, and enhance the St. Louis region as a world center for plant science. There's about 20 to 25 different faculty members at the Danforth Center. So we're basically structured similarly to a traditional academic biology department, but we're all focused on plant science. My lab is one of the labs at the Danforth Center. And as Barbara said, I started my lab in 2013. It's grown quite a bit, and this is the group that I get to work with now. It's a really fantastic group of people. I'm not obviously going to be able to talk about everybody's work today, so let me just highlight a couple of people who are, I would say, the main stars of today's talk. I'm going to talk about a lot of work from Kira Veely. She's a senior research scientist in my lab and works very closely with Greg Jensen, Kiki, Marisa Yoder, and Kiana Elliott, who's a graduate student in my lab. I'll be featuring a fair amount of her work. I'm also going to talk about work done by Dan Lin, who's a postdoc shared between my lab and Jim Carrington's lab, as well as Ben Mansfeld, another postdoc in the lab. And I also want to highlight that the work that I'm going to talk about today is, some of it is from my lab, led by my lab, but a lot of it is from other labs. We're really lucky to get to collaborate with a lot of different people. Particularly Nigel Taylor, Jim Carrington, Blake Myers, and Steve Jacobson, and all of the members of their labs. And then we also collaborate really closely with Kirk Zimick, who runs the Advanced Bioimaging Lab, Jeff Berry, who's a staff statistician at the Danforth Center. And also I'll be talking about some work from Ahuma Ogwanu, who's a scientist from Nigeria who spent about 9 months working in my lab. So what I'm going to try to cover today is I'm going to tell you about cassava. I'll introduce it as a very important crop and try to cover some of what I see as the opportunities and obstacles facing this particular system. I'll then dive a little bit deeper into one way in which we're using genome editing, and that's to address host pathogen interactions. And I'll give you two different examples of diseases where we have learned how pathogens cause disease and use that knowledge to use gene editing to create novel strategies for resistance. I'll touch briefly on some other traits, including herbicide tolerance, and then I'll wrap up with some unpublished work that will hopefully be submitted very soon, but is quite exciting, where we've actually been able to edit the epigenome. So with that, let me introduce the other main star of my talk, cassava. Cassava is an important staple food crop for millions of people around the world. It is primarily prized for these large storage roots that form under the surface of the ground. And those are dug up and processed and eaten much like a potato, so either boiled or fried or ground up and turned into something called gari. I have seen cassava chips more and more frequently, places like Trader Joe's, and it's also a component of tapioca. I have had cassava beer before. That was pretty tasty. As a plant, cassava grows as a very large bush, and it does produce seeds and flowers, but for the most part, cassava is propagated through cuttings. This is just this big bush cut up into stakes and planted in the ground, and the part under the ground grows new roots, and the part above the soil grows new shoots. And that part of its agronomy will have some important implications throughout my talk. Now, although cassava can be a very productive crop, my postdoc, Ben Mansfeld, pulled data from FAO Stat and graphed it here to show the point that while cassava can reach these really high maximum yields, many parts of the world that are very heavily cultivating cassava do not reach those maximum potential yields. Ben also took a look at the global median yields over time and saw that there has been some really impressive increases in crop yields during certain parts of our history. And what I'm going to try to convince you about today is that genome editing is this really interesting opportunity where hopefully we can solve some of these problems and increase those global yields even further. And what we're showing you here is that it was only actually, well, about a decade ago now, when the first genome was released. And so the hope is that that resource and a lot of the other genomics resources that are coming online for crops like cassava are going to speed this up. Now, most of you are probably familiar with teosinte, which is the wild relative of maize. And I like to use this slide to highlight the fact that the process that took teosinte into what we think of as modern day corn was thousands of years of breeding. A lot of traits were developed and tinkered and fine-tuned. In retrospect, or in comparison, cassava is really relatively wild. Some of the recent resequencing efforts that we've been involved in have shown that most of the varieties of cassava that are considered elite varieties of cassava are really only a couple of generations removed from their wild relative. So I think that means that there's a lot of room for improvement. We have reason to be optimistic. What I, again, will try to convince you of today is that genome editing can really help speed up that process. But I also like to show this slide at the beginning of my talks to highlight the fact that crops like cassava also have a lot of opportunity for discovery. This is a picture I took of cassava growing in a field in Tanzania, and I was completely blown away that it could be productive in this type of ecosystem. This is a cassava plant growing without exogenous fertilizer, without irrigation, and yet it's still able to produce those big storage roots underground. And I think we can learn a lot from crops like cassava if we're clever enough. So just to drive that home, one of the things that cassava does particularly well is drought tolerance. So I think genome editing can assist in the discovery of the traits that underlie that phenotype. And then there are a lot of different opportunities, I think, for further improvement with diseases, flowering, weeds, nutrition, and probably a lot else. So before I get into the actual ways in which we've used genome editing in my lab so far, I'd like to tell you about work that we've done over the last 5 or 6 years to develop all of the tools that we need to really be able to effectively do genome editing in cassava. One of the things that I'll tell you about is a long-term project in the lab to generate a really high-quality genome assembly for a farmer-preferred variety of cassava. I'll also touch on transformation capacity and then also on targets. So in terms of genome, I want to just emphasize quickly that plant genomes are very diverse, and each one is its own special butterfly, so to speak, to reference an earlier talk. Here we have Arabidopsis. It's a relatively easy to work with plant. It has a very small genome, only 5 chromosomes. It's self-pollinating, which means that although it is a diploid organism, 2 copies of each chromosome, those 2 copies are very similar. And so when we do something like sequencing the Arabidopsis genome, and we get all of those reads back, it's relatively easy to collapse them down into a single haploid representation of that genome. Maize is an excellent outcrosser. It has definitely a much more complicated genome, 10 chromosomes. But there's been, as I've already mentioned, a lot of effort going into maize. We have really great inbreds to work with that give us hybrid technology, but in the context of genome assembly, also allow those diploid chromosomes to be collapsed down into a single representation. Here's a picture from two of my favorite plant scientists, China Lundy and Sarah Hake, highlighting just some of the amazing genetic diversity that is present in this plant. Now, cassava, before I even really knew what cassava was, I was told that it was highly heterozygous. But I think at the time we didn't really understand what that meant. We didn't understand all of the different ways that a genome can be highly heterozygous. So over the last many years, my lab, led by Ben Mansfeld, have been working to create a phased genome for the cassava genome. So what I mean by that is cassava is also a diploid organism, but for each chromosome, we have two different haplotypes. And unlike Arabidopsis or inbred maize lines, the two haplotypes for each copy of the chromosome are really different from each other. And this is not only very confusing when we try to assemble that genome, but it's important for genome editing. If you're trying to design a guide RNA or a TALEN to target a certain sequence, you want to know if there's going to be polymorphisms between your two different chromosomes. So what Ben was able to do, working with a bunch of collaborators, was assemble both haplotypes of the cassava genome for all 18 chromosomes. And when he looks at them on sort of a high level, we see really nice collinearity, as one might expect, for the haplotype A and haplotype B of each chromosome. So that's what's shown here. But when you look closer, he saw that there is actually approximately one SNP every 70 base pairs between those two haplotypes. That's a huge amount of heterozygosity. That's similar to two different ecotypes of Arabidopsis, for example. The other thing Ben did is he took a slightly wider view and looked for relatively small but bigger than a SNP structural variations. So here defined as 50 to 50,000 base pairs. And somewhat surprisingly, he saw a whole lot of those as well, more than 5,000 structural variations between the two different haplotypes. Now there's a lot more in this paper. I encourage you to check it out if you want to know more about that. But what I want to emphasize in this talk is that in addition to being considered highly heterozygous, cassava is often talked about in terms of its high genetic load. And what that means is that things like breeding are very complicated because you end up homozygosing deleterious mutations. This was looked at more carefully by work from Ed Buckler's lab, as well as several other people, where they tried to actually quantify where are those deleterious mutations and how frequently are they? And we think the reason that cassava has such a high genetic load is in part because it is propagated through cuttings. So these spontaneous deleterious mutations arise, and then they're not purged through meiosis the way some other crops would be. So perhaps genome editing, if we get good enough at it, could be a way to potentially fix some of these deleterious mutations, and that would really help the efforts of breeders. So the next tool in our toolbox that I want to just touch on somewhat briefly is transformation capacity. Now in cassava, we are relatively lucky that we have fairly good transformation pipelines. And this is in large part thanks to two of my collaborators, Nigel Taylor, who has his own lab at the Danforth Center, and then a former member of his lab, Deepika Chauhan, who I got to work with a fair amount. And they, along with a lot of other people, have established this really pretty robust transformation pipeline. It's based on starting with immature leaf explants, inducing embryogenesis through tissue culture, and then using agrobacterium to deliver tDNAs and selecting through tissue culture. This process takes about anywhere between 8 to 12 months to go from your starting material to having this cute little regenerated plantlet. Now, one of the things that Deepika did several years ago was try to develop these transformation technologies for a bunch of different farmer preferred varieties of cassava. I mentioned that breeding is particularly difficult for cassava. And so we were thinking, if we really want to be able to do this gene editing and make a product that anybody would ever want, we need to do that directly in the varieties that farmers like. So Deepika wasn't 100% successful, but through a lot of patience and a lot of skill, she was able to develop transformation protocols for almost all of the different varieties that we were targeting. The other thing that we learned really early on with gene editing is that it's possible, because of this transformation system that we have, to get chimeras. John O'Dipia was a graduate student working in Nigel Taylor's lab, and he did some of the really pioneering initial work with CRISPR-Cas9. And he started out trying to target PDS, which we also heard about earlier today. And what he saw was, compared to wild type plants that develop normally in nice green leaves, he was able to make gene knockouts using gene editing and knock out that PDS gene. But he also saw he got a fairly high percentage of chimeras coming through. And thinking about why that might happen, it's pretty obvious in retrospect, I think, but maybe surprising before we had seen this result. You start out with an initial cell. That cell is infected by agrobacterium. It gets its tDNA, and then the actual editing is a secondary process that happens. That can happen in that original cell, or it can happen in subsequent cell divisions, and that's what leads to these chimeraisms. I think this is really important because this phenotype here, it was very easy to observe that chimeraism and know what's happening. A lot of the other traits that we want to be able to genome edit, it's not going to be quite so easy unless we actually look at it through sequencing or some other technique. So now I'll switch gears a little bit and tell you about some of the targets. I'm going to focus in on diseases first, because that's what my lab works primarily on. And before I do that, I need to give you just a quick crash course in host pathogen interactions and introduce how do pathogens cause disease and how do hosts protect themselves. I like starting with this slide because it's one of the things that made me originally fall in love with this topical space, which is that microbes induce these absolutely crazy morphological changes in their plant hosts. On the far left here, I'm showing you a tulip infected by a virus that's creating this beautiful streaking pattern on the petals. In the middle, I'm showing you a bacteria that's infected this echinacea plant and made it just absolutely confused about whether or not it's trying to produce leaves or flowers. That's called phyllidae. And then on the right, I'm showing you a pathogen of corn, a fungus, Staskawicz, that's creating these very large projections coming out of the corn cob. And in all these cases, the pathogens are using a very, on a high level, similar mechanism in that they express effectors to manipulate their hosts. This is true for bacteria, viruses, fungi, oomycetes, nematodes, and other insect pests. Here I'm cartooning a bacteria, but it's true for all of these different classes of pathogens. In the case of the bacteria, it's able to directly inject its proteins into its plant host through the type 3 secretion system. And some of these effectors will do things like shutting down plant innate immunity, things like inducing susceptibility genes. And so here we find another commonality in all of these different pathosystems. Most pathogens require host susceptibility genes to complete their life cycle. And in acknowledging this, we can see that these interactions could potentially be blocked through genome editing. So with that, I'll tell you two different short stories of how we have done this in my lab. The first one has to do with a potivirus that requires interaction with host translation machinery for viral protein translation. And the second story will be on a Xanthomonas bacterial pathogen that induces sugar transporters to promote susceptibility. In both cases, the stories are going to start with work done in model systems, and that's something that I want to try to emphasize throughout my talk today, the importance of this foundational research. Nobody really wants to do discovery work in CASSAVA unless you absolutely have to. So this was work that was done by Jim Carrington's lab a while ago now, where they took a potivirus of Arabidopsis and did a mutant screen for plants that were resistant to that virus. And what they found was that null mutants in the EIF iso4E gene were resistant to the virus. So on the far left is a wild type Arabidopsis plant infected with the virus, and you can see it's basically dead. In the middle is that EIF iso4E mutant doing quite well in the presence of the virus, similar to the wild type plant without the virus. And what the EIF iso4E protein is it's a eukaryotic initiation factor that the virus ends up having to interact with in order to translate its own proteins. There is a similar disease of cassava, also caused by a potivirus called cassava brown streak disease, and there's no genetic resistance available for this this disease. And so we thought, well, maybe this virus also has to interact with members of the EIF ISO4E family. So that idea launched a really productive collaboration between Jim Carrington, Brian Staskawicz, and myself, and was led by Michael Gomez, who was a graduate student at the time in Brian's lab, and Dan Lin, who's co-supervised by Jim and I. And our first question was simply which genes to target. So in Arabidopsis, they had discovered this EIF iso4E gene based on a mutant screen, but there's two other EIF4E family members in Arabidopsis as well. EIF4E and NCBP, which stands for novel cat binding protein. Like all gene taxonomy, the naming here is a bit atrocious. I don't take responsibility for it. In CASSAVA, our situation is a little bit worse. We have 5 different potential genes that the virus could be interacting with. So what Dan and Michael did is they took 2 different approaches. Michael worked on a yeast 2 hybrid, expressing the VPG protein from the virus with each of those CASSAVA family members. And what he observed was maybe a little bit of interaction with the iso4Es or the 4E family member, but definitely the strongest interaction with those 2 NCBPs. Dan took a slightly different approach, trying to look a little bit more in planta. He expressed the VPG protein and then co-expressed that in benthy with the different CASSAVA family members. And it then did co-immunoprecipitation. And he actually saw interaction with all 5 family members with the system. So it just so happened that at the same time, we were trying to target all of these different proteins with genome editing. And a double NCBP mutant line was one of the first ones that came through our pipeline. So Dan went ahead and challenged that line. And very happily, we saw really nice phenotypes where challenged the plants, let them grow up, and then compared to wild type plants, which developed that really striking necrosis in the roots, the double mutants seemed to be pretty symptom-free. This was quantified and seemed to be statistically significant, and then also correlated with a reduced virus titer. So these results were really promising, but we don't think this is full resistance. And so what Dan is working on now is generating higher order mutants. If we knock out all 5 members of the NCBP, EIF4E, EIF iso4E family, we're going to have a dead plant. Right? The plant needs to translate its own proteins as well. But perhaps if we knock out 3, we'll get slightly better. He's also trying knocking out just the iso4E clade to see how that compares. And then he had some other ideas as well. So the next story I'll tell you about has to do with a different class of pathogens, a bacterial pathogen, which induces sugar transporters to promote susceptibility. In this case, it's a Xanthomonas pathogen, and the model system that I'm going to tell you about that led to the gene editing in cassava is actually a crop too. So this is rice. It's of course a very important crop, and it happens to be susceptible to a Xanthomonas pathogen. In this case, the Xanthomonas pathogen introduces its own proteins into the plant cell, and one of those proteins is known as a tau effector. There's a bunch of different tau effectors, but a theme that has emerged from a lot of different studies between Xanthomonas and rice, as well as other systems, is that these tau effectors travel to the plant cell nucleus, bind to promoters, and upregulate expression of the downstream gene. And in a lot of these cases, those targets are susceptibility genes known as sweets, because they pump sugar out into the apoplast where the plant or where the bacteria is residing. Now, some earlier work in this pathosystem showed that it is possible to just knock out the sugar transporter, but that you often get a deleterious phenotype associated like sterility. So that doesn't seem like a very good strategy for crop improvement. Other studies have shown that there are natural rice alleles that show polymorphisms where that tau effector binds in the promoter that escape the binding by the tau effector while allowing the plant to still use that gene when it wants to. So that led to at least 2 different observations. One, a lot of follow-up studies showed that induction of sweets seems to be a very common strategy by these Xanthomonas pathogens across rice, pepper, cassava, cotton, citrus, and a number of other systems. And also this idea that in plants that don't necessarily have these natural promoter mutations that escape the binding of the tau effector, perhaps we could engineer edits into that, what we call an effector binding element upstream of the susceptibility gene, that would block the ability of the tau effector to bind and lead to resistance. One of the first proof of principles studies was almost a decade ago now at a Bing Yang's group, did just that. And they used the genome editing technology that was before CRISPR-Cas9 called TALENs to target that tau effector binding site in the rice genome and obtain a whole bunch of different mutations of that sequence. What they saw was that indeed the lines that they regenerated that had mutations that prevented the tau effector from binding showed enhanced resistance to this pathogen. Now, just a fun little aside, these talons are actually derived from tau effectors based on the code that tau effectors bind to DNA. Scientists re-engineered those into genome editing technologies, and it's a fun little circle of technologies and discoveries. So back to cassava. As I mentioned, cassava is susceptible to a Xanthomonas pathogen, very similar to the rice system. This is a picture I took in Uganda a couple of years ago before the world was locked down in a pandemic of some of the early symptoms that we see in this pathosystem. So you can see these classic, what we call water-soaked spots occurring on the leaves of this cassava leaf. And work that I did when I was a postdoc with Brian Staskawicz, collaborating with a graduate student in the lab, Megan Cohen, we discovered that similar to rice, the Xanthomonas pathogen of cassava injects its own proteins into the plant cell. And some of those proteins are tau effectors, for example, tau20, that go to the plant cell nucleus, bind to sequences, and upregulate sugar transporters. So that is very similar to the rice story that I've already told you about. Now, it is a little bit harder, even though we do have a relatively robust transformation pipeline in cassava. It's not quite as fast and easy as rice. And so we decided we did want to try to make mutations in the tau effector EBE, but we decided we would try to be as efficient as possible in that process. So Kira Vili and Ahuma Ekwanu began this work, and they decided that they would make one construct, but with 2 different guide RNAs, with the hope that with that one construct, they'd be able to recover an allelic series of mutants. This work has turned into the dissertation work of Kiana Elliott, and she's been characterizing these lines that have been coming out. So the strategy here was to have one guide RNA that targeted the tau20 binding site and another strategy that targeted downstream of the ATG. And we've started regenerating lines and have found a couple that have already been quite interesting, including one that deleted the whole region. And when Kiana challenged that with the pathogen, she saw significantly less water soaking. This is a close-up image of that initial water soaking spot that we use as an indicator of disease susceptibility. We also recovered a line that has a frame shift, a mutation upstream of the tau binding site, and then a frame shift mutation downstream of the ATG, which also seems to have reduced water soaking. We haven't yet recovered exactly the edit we want at the tau20 binding site, but Kiana has just obtained a bunch more lines and is in the process of characterizing those. So we hope still that we will be able to obtain a very specific mutation just in the tau20 binding site. So we'll continue searching for the EBE mutants, but we're also starting to think a lot about trying to target multiple genes at the same time. You can see that there is still some water soaking accumulating here, definitely less so than wild type, but not complete resistance. We know that these bacterial pathogens inject a lot of different proteins into their plant hosts. And so we predict that if we're able to knock out several genes simultaneously, we'll get stronger phenotypes. So I'm gonna leave the wonderful world of disease just for a few minutes here and tell you about some of the really early work that we did with genome editing focused on herbicide tolerance. This was a project that I became part of like the first thing I did when I got to the Danforth Center is join this team. It was led by Dan Voitas and also involved Nigel Taylor. And our goal was to develop herbicide tolerant cassava. This was before CRISPR-Cas9 was widely used. So we started out using talons. And I will just walk you through some of the key take homes from this project. So first, when I first heard about this project and was asked if I wanted to be part of it, I wasn't really sure whether or not herbicide tolerant cassava was something that was particularly important. I also just didn't know a whole lot about it, being from more of the disease world. But as I learn more, I have definitely gained an appreciation for how important weeds are as a major agronomic constraint on cassava cultivation. So weeding takes between 50 and 80% of the total labor budget. It's primarily done by women, and also a lot of farm children between the ages of 5 and 14 that have to miss school. For this reason, weeding is considered among cassava farmers as a major factor preventing upward mobility. The other reason that I think this is actually a happy coincidence that we ended up focused on this trait first is because tolerance to herbicides is an ideal phenotype for developing genome editing technologies, because we can work with that phenotype in tissue culture, as I'll show you. So our goal was to introduce 2 amino acid changes in the EPSPS gene to confer herbicide tolerance in a farmer-preferred cassava variety. Our original strategy was just to make those mutations that are located in the second exon of this gene. But early work that we did using transgenics told us that the native EPSPS promoter wasn't going to be strong enough. It wasn't going to be able to drive expression of that resistant allele at levels that would really protect the plant from the herbicide. So we realized that we were going to needed a new strategy, and that we could use, in theory, a repair template to replace the promoter, the first and second exons. The challenge here is that homologous recombination plants is incredibly rare. So it does happen, but it's very, very rare compared to things like yeast. So this was where the ability to select for the very rare event in tissue culture became very important. So Deepika spent a lot of time developing protocols for selecting herbicide tolerant events in vitro. It's no problem to start out with millions and millions of cells and then put those cells onto media containing that herbicide and select just the things that survive. And that's exactly what we did. We are able to regenerate a couple of plants that had successfully swapped in this 4.2 kb repair template that had a strong promoter, as well as the desired mutations in exon 2. And the results were quite good. The plants, once they were regenerated, showed really nice phenotypes in the greenhouse after application of glyphosate. So as I said, the herbicide tolerance wasn't really my area of specialty, but I feel like I learned a lot through that project. And one of the things that I came away from it really thinking about is, well, what could we do to overcome the limits of homologous recombination in plants for other traits? For example, I had really wanted to develop a tool to study cassava bacterial blight, the disease that I've already told you about. So I thought it would be cool if we could tag ME-sweet10A with GFP. It's not normally expressed in leaves, but if we had a plant that had GFP fused to ME-sweet10A, then when the pathogen was spreading through the plant, we would see GFP turning on as a reporter of the disease progression. The challenge is that ME-sweet10A is also not normally expressed in the tissue culture cells. So at the time, that was when Ahuma Okwanu was working in my lab, and she had this really creative idea of, well, what if we could just express TAL20 in those FEC cells as well? And so working with Kira, they did exactly that. So Kira designed this large construct that had the CRISPR-Cas9 reagents, had homology arms flanking the downstream side of ME-sweet10A with GFP in the middle. And then another component in the tDNA that had a promoter that we cloned from cassava as being expressed pretty specifically and quite highly in the FEC cells fused to TAL20. This large tDNA was introduced into cassava cells and then visualized under the microscope, and we were able to go in and cut out just those cells that were really expressing that GFP strongly. We regenerated plants from those few cells where homologous recombination we thought had happened. And indeed, we're able to show that in the lines that we regenerated, we got ME-sweet10A induced if there was TAL20 present. And then perhaps most convincingly was when we actually looked at the plants infected with the pathogen and indeed saw that GFP expression. So we're working right now with like Maya's group to try to generalize this tool for other traits and other phenotypes. And that's what we call the Surefire HR project. So the final story I want to tell you about now in the last bit is some work that I've been working on, gosh, for 6 or 7 years now. It's a collaboration with Steve Jacobson and Jim Carrington. And our goal is to try to develop targeted epigenome editing. Why would we want to do that? So I've told you about this. I've told you a lot about this ME-sweet-10A gene. I've told you that we could, in theory, just mutate the actual gene, but that is probably not a good idea because we know this gene is expressed in flowers. We believe it's very important for flower development. So if we just mutate the gene, we're probably going to have a sterile plant. I've told you that we have successful removed the EVE sequence using gene editing, but we're pretty worried that removing a large chunk of the promoter and 5' UTR might also lead to that gene not being expressed appropriately in the flowers. I've told you that we're working on introducing more specific mutations, but for some reason that we don't fully understand yet, that's been a little bit hard with this gene. So another strategy that could potentially block Tau20 from binding to the promoter is to put methyl groups on the DNA there to methylate that sequence. And methylation is something that's been studied very extensively in Steve Jacobson's lab. He's at UCLA. And he does all of his work in Arabidopsis and specifically has really focused in on this FWA allele. This is a gene that in wild type Arabidopsis plants is methylated, and that prevents the gene from being expressed. In the FWA mutant allele, that methylation is stripped and you get expression of the protein in this late flowering phenotype. So as I mentioned, Steve's lab has been working on this for a while now, dissecting all of the different components that maintain and establish that methylation. And a couple of years ago, a talented postdoc in his lab, Javi, who now has his own lab in Spain, demonstrated for the first time that it was possible to do de novo methylation using components of the RDDM pathway fused to DNA binding proteins. So here's a cartoon of the RDDM pathway. In this case, the protein that Javi used was DMS3, and he fused that to a zinc finger that bound to the promoter of FWA and was indeed able to observe methylation at that site. And I should mention here that we've been working with Steve and his group for 6 or 7 years now, and it's just been such a fun collaboration, partly because they're continually developing new technologies and just sending them to us to test in CASSAVA. And so while they've been doing all of this in Arabidopsis, we've been able to test them out in CASSAVA at the same time. And just recently, we've gotten some really exciting results. So this is work led by Kira Vili again. And what she did is she took that DMS3 gene and fused it to a zinc finger that targets the ME-sweet-10A promoter right where that EBE is, where the effector binding element is. She was able to demonstrate that recover several different lines that were expressing that construct and demonstrate that the protein was expressed. And then when we did bisulfite sequencing of the promoter, all of the controls are completely methylation free, but all of the lines that have the zinc finger DMS3 fusion, we see really nice methylation accumulating. Here you can see 0 to 100%. And these are the 2 CG contexts within the EBE of ME-sweet-10A binding site, showing almost 100% methylation. So here we've been able to demonstrate that we can indeed target methylation to where we want. So Kira's next question was, does that actually prevent Tau20 from binding and inducing ME-sweet-10A? So Kira worked with Greg Jensen to do this qRT-PCR, and indeed what we see is that methylation does prevent induction of ME-sweet-10A. So these 2 lines here are the ones that have the methylation, and we basically see no induction at all compared to the wild type plants inoculated with ZAM. And so then the final question, of course, was, does that actually lead to resistance? And again, very excitingly, the answer was yes. So here I'm showing you those water-soaked symptoms again, where we see the wild type ZAM and wild type CASSAVA leading to really strong water-soaking phenotype. the DMS3 zinc finger line showing significantly less water soaking. And it was interesting that the phenotype that we see here was very similar to a Tal20 knockout, suggesting, as we had predicted, that this is really the primary and main important target of Tal20 in cassava. So with this, we believe that we have established epigenome modification as a viable strategy for crop improvement. I think there's a lot of different potential applications for this technology. Kiana Elliott has been working hard developing different methods of quantifying these disease symptoms that we're seeing. So here's some of her work with ImageJ, just showing that it is a statistically significant reduction in that water-soaked area that we see. So as a final slide, before I open it up for any questions, of course, we're very interested to know what's going to happen when we do crosses. So we have all of those cassava lines, as well as a number of other DNA methylation technologies that we're working on in Hawaii now. And we're working with our collaborators, Joanna Norton and Sharon Wages, to conduct crosses. So we really need to know, is the methylation maintained in the absence of the transgene? And is that methylation heritable? And then similar to the traditional genome editing strategies that we're pursuing, we're curious, can we achieve a stronger phenotype by methylating several susceptibility genes simultaneously? So it does seem like genome editing and epigenome editing work well, and that's very exciting. So I'd like to close today with this final thought, which is that at this point, I really think the challenge is finding additional targets to modify for desirable improvement traits. I have a lot of people coming and wanting to work with me to make a better cassava plant, to make a better cotton plant, to make a better sorghum plant. And so I just want to really plug that I think this foundational research is what really allows us to do this work. We need to discover those traits so that we can know how to use our gene editing and epigenome editing technologies. So with that, I'd like to thank my lab again. Hopefully I acknowledged everybody as I went through, and I'd be happy to take any questions.
Thank you, Dr. Bart. It was very impressive, your work. And as a native Brazilian, I'm a big fan of cassava.
And so do you call it cassava or yucca?
I'm sorry, can you repeat that?
Do you tend to call it cassava or yucca?
Cassava, yeah.
Cassava, okay. Yeah. I'm always being told, oh, it's not, it's called manioc, or it's called yucca, or it's called cassava.
Yeah, we can.
That's a lot of names. Yeah,
that's true. And I guess I will open now for any questions. There is one question in the Q&A. So Marco Molina is asking, which cassava cultivar are you using for transformation? So we have primarily used 60444 in the past, but we've moved away from that. That was one of the varieties that was originally developed for transformation. Right now, mostly we're using TME 419. One of the things that we're thinking a lot about with the lines that we have in Hawaii right now is that we probably should try to recreate some of these epigenome modifications in a different variety, because cassava does a lot better at producing progeny if you cross two different varieties. So if you try to self two TME 419 lines, you're going to not really get very many progeny, and they're not going to be very healthy. But we have seen with some of our other collaborators that if you have two more distantly related cassava plants, you can get a large number of F1s. Okay, and Marco is also asking if you are able to segregate the backbone of the transformed plants.
Yeah, that's a really good question too, and something that we hope we will be able to answer soon. So at this point, we have a number of different lines there, and we just recently did sort of a low-level whole genome resequencing on those lines to quickly identify where the transgenes are. in one of the lines, the transgene is inserted on the same chromosome as the ME-sweet10A gene, and so that will make it harder to cross that transgene away. But we have a number of other lines now that have their tDNAs inserted on other chromosomes, so I'm feeling pretty optimistic that we'll be able to cross it away. And then the big question will be, without that transgene there, is that methylation inherited? Is it maintained?
Okay, and Pedro is asking if you are working off CIET, CGIAR, or if you plan to work with them given the CASSAVA collection that they have.
Yeah, this is amazing. We have a lot of CASSAVA aficionados on the call. So yeah, I do work a little bit with CI. They were kind enough to send us a bunch of wild accessions a while ago, which was really gracious. That was a ton of work on their part, just trying to screen more distant related varieties of manihot to see if we could find additional sources of genetic resistance. With the CGs, we do have at the Danforth Center pretty good connections with a number of them. We have potential talks. We have other projects going on where we have field trials going on in Africa, Uganda and Kenya, but it is a really big world and we're always very eager for additional connections. So if you feel like sending me an email afterwards and making any introductions, please feel free. Always happy to meet new collaborators and colleagues in the CASSAVA world.
Very nice. And one of our committee members, Mason, wants to ask a question.
Hi, Dr. Bart. Thank you very much for the presentation. It's really amazing, all the awesome genetic work you've been able to do in CASSAVA. So my question is, I thought it was really exciting. Oh, 1 second. Do you mind turning down the mic? I'm just getting some bad feedback. Okay, sorry about that. So yeah, so it's really cool, the approach that you had mentioned in being able to use, can you mute yourself? Okay, thank you. Okay, sorry about that. Once more. If you, yeah, looking at the work that you talked about in terms of increasing disease resistance by looking at the way that the effectors are interacting with those native proteins. So I was wondering, because you had mentioned that there could be a concern that, you know, if you do too many deletions of a gene or too many copies, that can lead to lethality. And so this is somewhat of an outlandish question, but with gene editing, you kind of never know. I was wondering if there has been enough description of how these effectors interact with those native proteins, and if a more kind of targeted mutagenic approach could be taken where those proteins are able to retain most of their native function while being able to more or less evade binding or interactions with those effector proteins from pathogens. So kind of an outlandish sort of idea, but was just wondering if there's any opportunities to kind of do more modeling and look into those interactions.
Absolutely. I don't think that's outlandish at all. Dan Lin actually wrote a NIFA postdoctoral fellowship on that exact topic for the EIF isophores. And so he's been doing some really great work with that, seeing can he do exactly what you said, which is to make mutations that would abolish the binding with VPG while still maintaining the translation functions of those proteins. So he's got some pretty exciting results, but I don't think he's ready for me to talk about them quite yet. So I won't, but stay tuned. And again, I think that's a great plug for how important this foundational science is. I would put that into the category of foundational science. It's stuff we can do with modeling or with model systems. Let's not try to discover that necessarily in CASSAVA unless there's a good reason to do it in CASSAVA. So good idea.
Yeah. Thank you so much for the response. And yeah, I'll be on the lookout for that work here in the upcoming years. Thank you.
Yeah, me too. He needs to write up his paper.
Yeah.
And we have one more question in the Q&A. Can you hear me? In some other plants, we see a positive correlation between disease resistance and transformability. So do you worry that one day the transformed plant cannot stack this resistance trait? And do you think these disease resistance traits can be stacked via traditional breeding, or do you think we will just modify the most up-to-date cultivars as we go?
Yeah, those are both really interesting questions. So let me answer the second one first because I think it's easier. The second question is whether or not it's better to gene edit something and move that into a breeding program, or just to continue to gene edit whatever plant you want. I think in cassava, there's a pretty good rationale for doing both, but because the breeding part is so hard, I think if we can make that ideal genome edit or epigenome edit that we want, then that can become a product because it can be maintained through clonal propagation. Especially with the epigenome modification, how that's going to be maintained through a breeding program. I think there's a lot of really, again, foundational science that needs to be done to really help guide how that will go. For the first part of your question, I think what you're referring to there, at least in part, is using agrobacterium for the transformation. And the agrobacterium is, of course, a pathogen in and of itself. And does that at some point get complicated? If I'm missing your question, feel free to write a follow-up one. But for that, if I do understand the question correctly, I would say there might be some concerns there. I can't think of exactly how, but it does make me think about a lot of the good foundational work that's going on right now with different plant systems trying to do direct delivery. We heard a talk earlier about carrot protoplasts and being able to do genome editing without actual integration of tDNA and through particle bombardment or these other mechanisms of introducing DNA. So that is something we would love to have in cassava, and just get away from the tDNA and the agrobacterium altogether. We'd also really love to get away from having to put our plants through embryogenesis, because we do get a ton of somatic weird stuff coming through the tissue culture pipeline, but we're just not quite there yet with cassava.
Brandon has a question. Go ahead.
Hi, Dr. Bart. That was a great presentation. I was just curious, you said that you saw a reduced water soaking phenotype when you had a methylation on the EBE site for Tau20. Have you seen, or are you concerned that the disrupting the interaction between the Tau effector and the EBE site may drive some sort of recombination effect within Tau20 to target an alternative site within a sweet gene?
Absolutely. So you're in Levo Sean's lab, right?
Yeah, how'd you guess?
Tell her I say hi. I think you
Hello.
mentioned that in one of your emails, but please tell her I say hi. So yeah, that's a great question. And I think that's absolutely what would happen if we deploy this as a product just out in the field. The pathogen will just mutate around that resistance quite quickly, I would imagine. I do think that that's an interesting, it's something that we talk about in my lab a lot, with how does genome editing of the DNA sequence compare to epigenome editing in that context? Would the pathogen have a more difficult time mutating around a broad swath of methylation? It would have to presumably mutate several different RVDs in order to have a new recognition specificity. So that could be a plug, assuming that it eventually works for epigenome modification. But the real answer, as I'm sure you know, is target multiple susceptibility genes and apply that as the product, not a single one.
Okay, great. Thank you.
So does anybody has any other question? Khushbu? Go ahead.
That was a very nice presentation, Dr. Becky. I have a small question for you. Have you explored the wild relatives for knocking out the sweet 10 gene? Like, instead of the expression for the with the TAL, the expression is getting stopped. So have you explored the wild relatives? If there is any different isoform available, or is it equivalent in the function?
So do you mean, have we looked for wild relatives to see if there's naturally occurring alleles that wouldn't be targeted by Tau20?
Yes.
Yeah, so we have looked at the diversity of cassava that we have access to that's grown and that we have in our germplasm collection at the Danforth Center. But I think that would be, going back to one of the earlier questions, a really great thing to do at a place like CIAT that has this immense, really amazing germplasm collection that's much more diverse than what we have at the Danforth Center. Sounds like a great collaboration.
Makes sense. Thank you.
Any other questions? Okay, so I have a question. Is the sweet genes that you identified, they are specifically targeted by Xanthomonas, one specific species of Xanthomonas, or they can be generally targeted by other species?
Yeah, so it's been interesting over the last decade or so to see that different species of Xanthomonas target sweet genes, but each in their own host. So if I took the Xanthomonas that infects cassava and put it into cotton, it won't cause disease in cotton. But the Xanthomonas that infects cotton also has tal effectors that also target cotton sweet genes. That's a very specific interaction between tal effectors and these sweet genes, but the involvement of sweet genes in general extends even beyond just Xanthomonas pathogens. So this has been, there's a paper, I think, from Wolframmer's group a number of years ago showing that a whole different diversity of different pathogens target in different ways sugar transporters as part of their pathogenesis. It makes sense, right? The pathogen has to eat.
Yeah, right. Okay, so I don't think we have any other questions.